Lucifer is a little non-degenerate genus of prawns. It has a long body but many fewer appendages than other prawns, with only 3 pairs of parapods remaining, all without claws. It also bears no gills. The female uniquely among prawns carry the fertilized egg on her pleopods until they are ready to hatch. The head is extremely elongated and bears long antennae and eye with a stalk. The fifth parapod is absent or vestigia. Luminescent organs are absent, but luminescent cells are present in Chelsen. Phylum Arthropoda, subphylum Crustacea, class Malacostraca, order Decapoda, and family Luciferidae. Phylum Arthropoda, subphylum Crustacea, class Malacostraca, order Decapoda, family Luciferidae. Mysis lava. Mysis is the larvae of prawns. It is a small shrimp-like plankton. It has characteristic thoracic body pouch and carapace. 2 anterior appendages are present. Auditory sac is seen on the entopodite. Gills are absent and respiration occurs through the lining of the carapace. It has 5 pairs of posterior thoracic legs which are biramous with flagellas, exopodate that help in locomotion. The abdomen is well elongated and well developed biramous pleopods. They are capable of more vigorous swimming and can seek out, capture, gasp, and hold food consisting of variety of 200 zooplanktons. Phylum Arthropoda, subphylum Crustacea, class Malacostraca, order Mysidae, family Mysidae. Lucifer, here we could see a picture. Antennae, uropod, pleopods, parapods. Mysis lava, rostrum, antennae, antennule, Mysis lava, cephalothorax, abdomen, Chelsen, uropod, and pleopods. Sagitta is a marine planktonic animal which lives throughout the world. It can swim very slowly and most of the time it floats on the surface of the sea. It has an elongated torpedo-like bilaterally swimming symmetrical body. The body is divisible into 3 parts: head, trunk, and tail. The head is more or less triangular in outline and is separated from the trunk externally by a constricted region called neck and internally by a coelomic septum. The head bears 4 rows of teeth, one pair of anterior teeth and another pair of posterior teeth. Head is covered over by a hood on dorsal side. The trunk is slightly broader towards the middle region and is provided with 2 pairs of lateral fins. The fins are thin and transparent extensions of epidermis of the body wall. Sagitta are bisexual. The ovaries and testes are paired structures. Sagitta is a bisexual organism. Phylum and then class Sagittaria, order Aphragomorpha, Sagittaridae. Sagitta hooks, head, eye, collarette, sensory papillae, ventral nerve ganglion, intestine, anterior lateral fin, trunk, eggs in ovary, posterior lateral fin, anus, testes, seminal vesicle, tail fin, and tail. Quantitative estimation of marine planktons. To quantitatively analyze the amount of marine plankton in the given sample, materials required: plankton counting chamber, cover glass, dissection microscope. Principle: marine planktons are important links in the aquatic food webs. Their occurrence and distribution influence pelagic fishery potentials. Qualitative analysis can be done by numerical method and a differential count method. Numerical method. Quantity of plankton is measured in terms of number per square unit volume of water sample. The plankton is counted in a counting chamber and total count of all marine plankton in the sample is counted in liter. So then differential count method. This is an enumeration of all or some of the different kinds of planktons by counting and recording the number of each individual of each species. Procedure: total count of planktons. This involves the counting of total number of individual plankton in the sample and expressing in 1 liter. Close the concentrate bottle of marine plankton and differential count. This is enumeration of all or some of the different kinds of planktons by counting and recording the number of each individual of each species. And procedure: total count of planktons. This involves the counting of total number of individual planktons in the sample and express in 1 liter. Close the concentrate bottle of marine plankton and invert it slowly until the concentrate get thoroughly evenly mixed. Pipette out 5 ml of concentrated marine plankton sample by means of wild bore glass tube and transfer it to test tube. Dilute the sample taken by adding 20 ml of water. Water added depend upon the concentration of plankton sample. Shake well and keep it on a bottle. Shake well and suck out 1 ml of plankton and transfer the same to the clean plankton chamber. A typical counting chamber is a glass square of 1 inch into 1 inch sealed into an ordinary glass or microplate that is 1 mm deep and with capacity of exactly 1 ml. 1 inch into 1 inch, 1 mm deep, capacity of exactly 1 ml. The plankton chamber, if glass is provided, mount the chamber until whole cell is covered, allowing the excess to overflow. In case any air bubbles are trapped, repeat the process again. To the plankton chamber, if cover glass is provided, mount the chamber until whole cell is covered, allowing the excess to overflow. In case any air bubbles are trapped, repeat the process again. And carefully transfer the counting chamber to the stage of dissection microscope and wait for a minute until the planktons are settled down. Now select a square with the plankton chamber randomly and count the number of planktons. Likewise, repeat counting in more squares. The counts are recorded in a tabular column and their average number is found out. Now shake the bottle again well. Again take a fresh sample in counting chamber and repeat the process as above mentioned and record it. Likewise, take 5 samples and total count is calculated. While counting, take care of the following. Each separate organism should be counted. Each filament, colony, larval stages, eggs or partial or as whole should be counted as one plankton irrespective of size. Count only after planktons are killed. Ignore all kinds of debris and other objects like skin filaments. Also avoid planktons which are partially inside and outside in position in the plankton chamber. So here we could see results, observations and calculations, plankton, Lucifer, first view, second view, third view, fourth view, fifth view, total. Okay, Lucifer, Sagitta, Mysis, Lucifer, Sagitta, Mysis, copepod, Crabazoia, and Ostracode, Amphipod, Mysis, total. Lucifer, Sagitta, Mysis, copepod, Crabazoia, and Mysis. Okay. Dilution factor 5. Dilution factor 5, sample taken for analysis 1 centimeter square. Count of total marine plankton in the given sample into dilution factor by total number of sample. Total count of marine plankton in the sample in liters into 5 into 1000. 68,000 organism per liter. Then differential count. Total number of Lucifer, total number of Sagittarius, total number of Mysis, total number of copepod, total number of Crabazoia, total number of Ostracoda, total number of Amphipoda, total number of Mysid. Dilution factor 5. Sample concentration taken for analysis 1 cc. Total number of sample taken 5. Differential count of Lucifer 8 by 5 into 1000 into 5. Differential count of Sagitta 6 into 5 into 1000 by 5. What is 5? Dilution factor 5. Sample concentration taken for analysis 1 cc. Total number of samples taken 5. Organism per liter like that. Okay. Then we will be plotting a graph with this. Differential count of plankton. Total number of planktons in 1 liter. 68,000 organisms per liter. Differential count of copepod. How to do this? Into 5 into 1000 by 5.